Okay, firstly, I'm very appreciative, and so is Paul, of the invitation to speak here. So thanks to Mary Lou and Ruben for organising and funding our involvement. It's been a wonderfully informative meeting for us. I'm just going to give just a very broad introduction, and then I'll pass over to Paul. I'll just talk about the avocado industry in Australia and the premise for the project. So the Australian industry is a rapidly growing industry. It's pretty well dominated by Hass, with a few other cultivars making up the difference. In Australia, it's, I guess there's a lot of Australia that is suitable for avocado production. Over 60% of the production is in subtropical Queensland. So that's from the Queensland border right through to Bundaberg. Bundaberg is about the latitude of the Tropic of Capricorn, is where the majority of production in Australia. There are other minor production zones in Western Australia and also in northern Victoria, southern New South Wales and other areas of the east coast of Australia. In Australia, the main pathology challenges are Phytophthora root rot. Because the conditions, especially along the east coast of Australia, are quite humid. Post-harvest diseases are a serious issue for the industry in terms of quality of fruit hitting the market. In particular, Colototricum gliosporoides complex causing anthracnose and stem end rot. We also have another soil-borne pathogen called Phylinus noxious, which is a rainforest decay fungus that is entering the avocado plantations and causing death of the trees, and there's no really good control for that. Avocado sunblotch is more of a biosecurity threat. It's a pathogen that has a limited distribution and is controlled by a clean planting material scheme. And laurel wilt was identified as one of the largest biosecurity threats to the industry. We don't have the beetle and the fungus in Australia, but through logic and extrapolation, if it can get to Florida, then it should be able to get to Australia, which is geographically closer to the origin of the two organisms. And we also have very large trade relationships with Southeast Asia, our largest trading partners. So that was the premise to the project, was to actually have preparedness for those two organisms and to develop diagnostic assays. So that's where two virologists came into the picture, because we had skills in molecular identification, and when there's a shortage of traditional taxonomic skills, molecular assays can come into the fore. As part of the justification for the project, we wanted to test the diagnostic assays against the endemic flora and fauna of Ambrosia beetles and fungi, and in the course of that study, we've looked at U. wallacea, which was identified at about the same time as the beginning of this project. So I'll pass over to Paul Campbell, who will elaborate. Paul worked as the actual scientist on the project, and we'll talk about what he did.
I'm going to be mainly talking about the two areas here. Go forward or backwards. So we had two areas that we found the wallacea, up in Cairns region, which is up here, and around the Sunshine Coast. So you're looking at about 1,000 miles between the two areas. Which way goes forward? There we go. So back in 2009, there was some reports in macadamia in the Sunshine Coast, some beetles boring into some trees, pretty much destroying the trunks of these trees. I think there was 10 individuals in the one thing. So They were sent off to Roger Beaver in Thailand and came back as Fornicatus. We thought, okay, very nice. And since then it's vanished. We haven't seen it in macadamia since. Then 2010, another grower in pretty much the same area, probably about 20, 30 miles away, just sent some branches around, said, you know, what's going on with these trees? We're getting some problems with their avocado. He was seeing these beetle boring holes into dead branches and wondering what was going on. So again, we sent it off and came back as Fornicatus. Then in 2011, we were up at Cairns for the Avocado Congress, and we were implementing this trapping regime to look for glabratus and whatever's out there in Australia, and just trying to tee up some places to put things out with the grower. And grower's like, yeah, well, I've got some trees that died with some beetle holes in them. Do you want to come out and have a look? So it may be more widespread than we know, but wherever we've gone looking, we've actually found it. Wherever I've, say, put out traps, even though there's no reports of these things, you look in the smaller branches and we've found the actual beetle. So it's actually quite hard to see in the canopy. I've got some pretty excellent photography skills. So it's this branch here, no leaves left at all. There's actually another one that runs back through here. You can see it probably runs up here. So you can just walk down the rows and you'll just see some branches, no leaves, dead. It's common, not that common. Probably in that row, you're looking at every fifth, sixth tree in this orchard has these dead branches. And you're looking at probably 1.5 to 2 inches max. We're not seeing it really in this area boring into any larger branches. But the main bore site, I think it's in the next one, is right in the base of the branch. So these two branches are still healthy, still full covered with leaves. And if I section through here, you can see the galleries and the discoloured wood. So it doesn't appear at this stage that the fungus is moving back down the tree. It's moving up and out. So, but then that could be just a timing thing as well. Like if I come back in another year, maybe those larger branches have died. But then there's probably going to be beetle holes in them. So is the fungus knocking the branch back and then the beetle's moving back in again or not? Once you look at that sort of smaller branch, there's probably, you know, every centimetre or so down near the bottom has beetle holes in them. So it's moving fairly rapidly, and as you can see, there's quite a bit of frass coming out at the base of the tree. In this spate, we don't see any of the, oh, actually I do. Sorry, I have to jump around a bit, it gets a bit confusing. So this is up in Cairns. This is the tree. The farmer's lopped most of the limbs off and he took the rest of the tree out not long after this. These trees were hit by a cyclone probably a year or a year and a half before. Took a lot of the canopy away, so they actually got quite badly sunburned. So, but this is in the trunk, so we see quite a lot of actual boring holes from this beetle. This is in Shepherd, by the way, it's not Hass. When you cut it back, you can see the beetle bore holes and some discoloration in the tree there. So in Cairns, the only region we've actually seen trees dying, but to make it a little bit more confusing, this is the same sort of symptom we see in Sunshine Coast. So this is in Cairns as well. So the smaller branches, the wilted leaves, and the bore holes, the branches will snap off at that sort of level. To make it a little bit more difficult, we actually found this Microperis species in those trees as well. So probably for every Fornicatus hole, there was probably 10 of these. And we've isolated some fungi out of these, and it's a Bionectria species. So what's going on in Cairns to kill the tree? Is it the Fornicatus? Are these moving in afterwards? Did the sunburn hit them enough that the Fornicatus moved in and then these moved in, or these moved in, the Fornicatus moved in? We're not really sure. So back to the Sunshine Coast. So this is probably, I got these about a month ago. I went back up to the growers, and that beetle hole is probably 3-4 days old, tops, and you can see the same exudate there from the boring in. This is Fornicatus, and I think I stripped it. Yep, so this is the same branch there. Tiny little discoloration here, and if you strip the bark all the way, you get this reddish stain. We're pretty sure that's just the natural wound response from the avocado. So the fungal ones, you get sort of a browny or a grey colour, where the natural polyphenols, you're getting that red stain all the way up. But I have managed to get the Fusarium isolate pretty much all the way up here. I got it up here, but not out to here, so towards the edge of the limit of the stain. And that's probably only maybe a week tops in that branch. So the fungus seems to be moving quite rapidly through the branch, but it doesn't seem to be moving back down into the larger branches and causing tree death. But if I strip an older branch, I didn't have a very good photo of it. You strip an older branch, you do get very big sectoring, so you get a very clear bit of nice white avocado branch, and then, so some of them, they say 3/4 round, were brown and black, and it extends from the tip of the branch back down towards the beetle holes. So this is what we got out. Fusarium species. I like to call them sort of chili shaped. I'm not really sure of the other ones. They, maybe Akeef will mention it. We sent some for him to do some typing with, and they apparently fall out somewhere between the Sri Lankan strain and the Californian strain. So it's probably a new species, but we're not exactly really sure what it is. Sorry, I typed the Fornicatus that we have in Cairns and the ones we had in the Sunshine Coast, and they're the same. There's 2 base pair differences in the COI sequence in that region. So either they are evolving very quickly. If they've only been here for a couple of years, they may have been here for a lot longer than we think, or we've had 2 different introductions of these things. But just from the EF sequences, the actual symbiont is the same between the 2 regions. So what have we got out of it? Mainly just Fusarium species for both of them. The Bionectria came out of that Microperis species. I've done inoculations for both of them on avocado and seen nothing for either of them at this stage. We might just have to wait and wait and wait. But we also get a Bavaria bassiana out of the ones in Cairns, and I didn't notice it when I actually took the photo, but we actually have one of the Fornicatus here that's been infested with the bassiana. So I had real trouble getting a lot of the Fusariums out of there because everything that came out of that log was infected with Bavaria. So it seems to be controlling it quite well in Cairns. You very rarely see any of them flying around. You'd see a lot of the Microperis, but really none of the Fornicatus. So we're actually doing some of the surveillance. I paid good money for some excellent help, but it didn't seem to work that well. So we made our own traps and scattered them around in Cairns, Sunshine Coast and the northern New South Wales area, coated with tanglefoot, and we baited them unbaited ethanol and manuka oil, which is pretty much the same as manuka oil, except we pronounce it correctly in Australia. Andrew gave me a little note to read out, but here we go. Manuka is a Maori name for tea tree Leptospermium, so it's manuka, not manuka. So 4 trapping locations in Queensland and New South Wales. And pretty much as you'd expect for Ambrosia beetles, we pulled out about 40-something species from the different areas. Most of them attracted to ethanol, except this one species here that was ridiculously attracted to the manuka oil. It's native. I think I've got a picture. There we go. Ambrosia compressus. It's a native species, and that was trapped in the orchard that we found the most Fornicatus in. So I don't know what it's doing there. Natural decay of the avocado wood in the actual orchard, but it's how it's running. So the other bit of the project's running a metagenomic analysis of what's actually out there. The whole idea is we don't have many good entomologists anymore, so we want to actually barcode all the beetles we find, put them in a collection, and actually have a look at what they're actually carrying. The idea is to even use them as little sweepers, just what's out in the environment. So I'm not really caring whether it's in the mycangia or not, whether it's in the gut or not, or whether it's even on the surface, even though I quickly surface sterilise them, because they're flying around, trapping whatever they can find, eating different fungi that they find on trees. So just an idea of what's actually out there in the environment. So we just ran a multiplex PCR for the 2 cytochrome oxidase regions in the beetle, just so we can know what's what, and ITS and the elongation factors for the fungi to try and get at least a good locus approach. And unfortunately, it's the data's been done by a Chinese company, and they're still fabricating my data for me, so we can't show you any results at this stage. So in Australia, we have the beetle, or maybe it's not the beetle, you guys can fight about it, and we have some sort of fusarium. We have dead avocado branches and some dead avocado trees, but we're not really sure what's actually happening for the whole lot, so thank you.